Hi guys, welcome to GeeklyEDU Biology. This is me Sona Singh, and today we are going to talk about in detail blood groups and the transfusion rules that we must follow and the logic behind that. Let's get on with it. We all know what blood is. It's a fluid that flows inside our bodies and it carries a lot of substances. So let's take one particular blood capillary. It has those thin endothelial walls through which the components of blood fuses out and provides our cells with everything. So inside this blood capillary, we have blood, and if you centrifuge your blood, you can find there are three peculiar portions. The upper yellow portion that we call the plasma is the fluid part, which carries your glucose, waste, and ions, and hormones, and a lot of sort of thing. The lower 45% is the red blood cell. You can see red blood cells are pretty eminent. They occupy a great deal, and just in between them around 1 to 2% is a buffy coat that you'll find, and that buffy coat is platelets and WBCs, which prevent bleeding, unwanted bleeding, and make sure that you stay healthy, you fight infections. But the blood groups that we are going to talk about is all about RBCs, red blood cells, that are present there in huge amount in our blood. So if you take one RBC and look at its surface, so it will be like a biconcave shape, somewhat I like to call it donut shape, which is almost devoid of most, if any, organelles are present. It's just ribosomes. But on its surface, you'll find these molecules, sometimes proteins, sometimes the carbohydrates. These molecules are called as antigens. Now, why I call them antigens? Because your immune system is reactive or can be reactive towards these. Now, each antigen comes in different flavors. So let's talk about the triangular shaped antigen here. So let's say by representing the differences by colors, I can say there could be a black triangle, there could be a red triangle, or there could be a blue triangle. Basically, three different versions of the antigen that I'm talking about. All right. Now, they are determined by alternative forms. So because they would be a carbohydrate or a protein, if there's carbohydrate, that would be synthesized by different proteins made by different forms. So what are blood groups? It's a way to classify blood based on what sort of these antigens you have. And there are 33 systems of blood classification systems, and we will study one of them, which is a chief of transfusion, that's ABO system. So two chromosomes play a very crucial role for the ABO system, that's chromosome 19 and 9. So chromosome 19 has a gene called flood gene, but right now we are just going to call it H gene. Gene will code a protein, which is an enzyme. So if we have the big H copy, we'll have a carbohydrate H synthesizing enzyme. If you have a small H copy, no enzyme, no carbohydrate. Now the chromosome 9 has another gene which we are just going to call as ABO gene. It has its own specific name, but ABO gene which can have three different flavors. If the flavor is IA, it will modify the H by adding N-acetyl galactosamine, and we call this as an AA antigen. If we have the flavor IB, it modifies it to form to add in galactose, and we call this a B antigen. And if we have a small I, it will have no function, and hence H will be left as it is. H here represents fucose. Now, so we are diploid organisms. We have a copy from a father, we have a copy from a mother. So we have two alleles, two forms. It might be we may have the same or different forms. So if I have IA, IB, IA, small I, as long we should remember IA and IB are codominant. What does codominant mean? That both enzymes will be present, both genes will be active. But if I have a small I is recessive, then only I have the A antigen. Now, it's important that you remember this. Similarly, if I say B blood group, what can be the possible gene or allelic combinations that you might be? So you might be IB or IB small I. All right. O blood group will only be there if you are small I. We have the recessive allele from both of your parents. Okay. Now, why does this matter, and how does this matter when we are talking about blood transfusions? So when you have an antigen, you also have an antibody against it. But your body kind of grows tolerant towards the antigen that you already have. It will not try to synthesize an antibody. So let's say you have an A antigen, your body will not make the antibody anti-A. But the other antigens that you come across by the surrounding, for example, there's a bacteria which has a similar looking antigen, your body recognizes it, and there is. So it makes antibodies against it. So if you're A, you'll have anti-B antibodies that will work against the B antigen if you ever were to receive a transfusion. Right? So A blood group has antigen A, B has antigen B, O has no antigens, and AB has both the antigens because they are codominant. So the blood with A antigen will not have anti-A antibodies, it will have anti-B. Similarly, the B will have anti-A, O will have both because it has got no antigens, and AB will have no antibodies. Now, if this guy, let's talk about a guy named Sam suffers an accident, and if he receives a blood transfusion from a donor, the blood will give 45% RBCs. Remember, the blood is 45% RBCs, and there would be antibodies against the antigens that are not in his blood, in his plasma. But you see, majority is antigen. So how we determine is your body should not hate the RBCs that you're receiving because then there will be a reaction. Antibodies is fine, it will simply dilute out. So you should not have antibodies of the same type of the antigen that you're receiving. Mind that. This is very important. Okay. So now let's say which people can serve as donors to A and O because they do not have the B antigen. And A already has anti-B antibodies, right? So because those will not have B antigens, they can easily donate, right? So incoming RBCs will not be attacked. That is the funda that you have to remember. The incoming RBCs should not be attacked. Now B can take from B and O, right? Because those won't be attacked. O can not take from anyone else other than the O. AB can take from anyone because it will not attack any gens. It doesn't have any antibodies. So I hope this makes sense, right? Now, before we wind up, let's talk about one more scenario. Remember, we have two possible alleles, big H, small h. Big H is dominant, and that is what makes the basal carbohydrate that can be modified by chromosome 9. But what if we have small h, no antigens? Possible because there won't be a basal carbohydrate, right? So this is a rare blood type, small h, first found in Bombay, and hence the Bombay blood type. This will show agglutination against all other blood types. Now this RH as well, do you want to know about it? Let me know down in the comments below.
We hope you like this video. Don't forget to press subscribe, give us a thumbs up, and stay geekly.